Once upon a time, there was a little double helix DNA molecule who lived in her cozy little nucleus. She was feeling very lonely and decided it was time for her to share her genetic codes she's been carrying for a long time. It was time for her to pass on her information to create precious proteins for her home cell. The first step for our little DNA was to transcribe her nitrogenous bases into magical mRNA. In order for her to do so, she had her fairy godmother DNA polymerase help her out. First, the DNA polymerase splits the little DNA in two, so you can see her template strand and her coding strand. Her template strand will be the most important part in creating the magical mRNA. A specific nucleotide sequence in the DNA called a promoter tells another friend, RNA polymerase, to begin transcribing. Nitrogenous bases for the mRNA match up to the corresponding bases on the template strand with the help of RNA polymerase, creating a nice little portable copy of the genetic code. It goes along the DNA's template strand, matching all the little bases until it is complete. The last step is the addition of a nucleotide cap at the 5' end and a poly A tail at the 3' end. This not only protects the RNA, but also helps facilitate translation. It is now time for the next part of the journey in our little molecule's life. This part is called RNA splicing. RNA splicing is when non-coding sequences of mRNA called introns are cut out and other regions called exons are joined together to create one cohesive mRNA molecule that will be translated into amino acids. The mRNA is now finished and is ready to continue on its adventure to leave the nucleus. In order for proteins to be made, the code must be read off of the mRNA and paired with the corresponding amino acids. Our little RNA has to make the long journey from its cozy little home in the nucleus to the ribosome, which is the site of protein synthesis. The 5' cap helps the RNA to bind to the ribosome and begin translation. The mRNA is read in groups of 3 bases. Every 3 bases is called a codon. Each codon codes for either an amino acid or a terminating sequence. Another type of RNA, called tRNA, reads each codon and assembles amino acids accordingly. This translation continues until tRNA reaches a stop codon. The protein breaks apart from the ribosome to be further processed and packaged. Our little friend, DNA, transcribed her genetic code into RNA that went on to be translated into a chain of amino acids and packaged and processed into precious proteins. The proteins made along this journey will be used for many, many different purposes within the cell. However, some errors can occur during transcription that can cause mutations and alter the entire process. Bases can be switched or deleted or added. A switched base can cause a missense mutation. This mutation changes the amino acid that gets coded for. The new amino acid may not affect the protein at all due to the sequence change coding for the same amino acid. This is called a silent mutation. The next type of mutation possible is called a nonsense mutation. A nonsense mutation is when the substitution of bases changes one codon to a stop codon. This results in early termination of the translation of mRNA.